Before start this video, please subscribe my channel and click on bell icon. Thanks.
Washington, researchers at Washington University School of Medicine in St. Louis have found that individuals with early-stage Alzheimer's disease, when brain changes have started but before cognitive symptoms are noticeable, have a variety of gut bacteria distinct from those in healthy individuals. The research, which was published on June 14 in Science Translational Medicine, paves the way for the development of microbiome-altering preventive treatments for cognitive decline as well as the possibility of using gut bacteria to identify those who are more likely to develop dementia. We don't yet know whether the gut is influencing the brain or the brain is influencing the gut, but this association is valuable to know in either case, said co-corresponding author Gautam Dantas, Ph.D., the Conan Professor of Laboratory and Genomic Medicine. It could be that the changes in the gut microbiome are just a readout of pathological changes in the brain. The other alternative is that the gut microbiome is contributing to Alzheimer's disease, in which case altering the gut microbiome with probiotics or fecal transfers might help change the course of the disease. The idea of studying the connection between the gut microbiome and Alzheimer's disease ca. ANSYS treats and studies people with Alzheimer's disease. Dantas is an expert on the gut microbiome. Scientists already knew that the gut microbiomes of people with symptomatic Alzheimer's differ from the microbiomes of healthy people of the same age. But, ANSYS told Dantas, nobody had yet looked at the gut microbiomes of people in the critical presymptomatic phase. By the time people have cognitive symptoms, there are significant changes that are often irreversible, said ANSYS, the other co-corresponding author. But if you can diagnose someone very early in the disease process, that would be the optimal time to effectively intervene with a therapy. During the early stage of Alzheimer's disease, which can last 2 decades or more, affected people accumulate clumps of the proteins amyloid beta and tau in Th. Dantas, ANSYS and first author Aura L. Ferrero, Ph.D., then a graduate student in Dantas' lab and now a postdoctoral researcher, evaluated participants who volunteer for studies at the Charles F. and Joanne Knight Alzheimer Disease Research Center at Washington University. All participants were cognitively normal. As part of this study, participants provided stool, blood and cerebrospinal fluid samples, kept food diaries, and underwent PET and MRI brain scans. To distinguish participants already in the early stage of Alzheimer's disease from those who were healthy, the researchers looked for signs of amyloid beta and tau accumulation through brain scans and cerebrospinal fluid. Of the 164 participants, about a third, 49, had signs of early Alzheimer's. An analysis revealed that healthy people and people with preclinical Alzheimer's disease have markedly different gut bacteria in terms of the species of bacteria present and the biological processes in which those bacteria. These differences correlated with amyloid and tau levels, which rise before cognitive symptoms appear, but did not correlate with neurodegeneration, which becomes evident about the time cognitive skills start to decline. Thanks for watching. Please subscribe my channel for more videos.